Welcome back to the Biology Domination podcast by Hidden Wisdom Academy Biology. I'm your host, and today is day 19 of our 30-day NEET biology crash course. We're talking about excretion, specifically excretory products and their elimination. Now, I know some of you might think this chapter is dry, pun intended, but I promise you, this is a goldmine of direct NEET marks. We're going to go deep today. Make sure you're taking notes, or just keep coming back to this episode as you revise. Let's get into it. Let's start with the basics. Why do we excrete? When proteins and nucleic acids are broken down in the body, they produce nitrogenous waste, primarily in the form of ammonia. Now, ammonia is highly toxic. It has to be diluted massively before it leaves the body. Aquatic animals, fish, aquatic invertebrates can afford this because they're surrounded by water. They are ammonotelic. Humans and other mammals can't afford to lose that much water, so our liver does something clever. It converts ammonia into urea, which is far less toxic and can be concentrated in urine. We are ureotelic. Then you have birds, reptiles, and land insects. They go even further. They make uric acid, which is practically insoluble, almost non-toxic, and needs almost no water to excrete. That's why bird droppings look like paste. They are uricotelic. NEET will give you an organism and ask which type. Know your group's cold. Now let's get into the nephron, the functional unit of the kidney. You have roughly 1 million of these in each kidney. The structure goes Bowman's capsule, then PCT, then the loop of Henle, then DCT, then the collecting duct. Blood enters the nephron through the afferent arteriole. It's wider. It flows into the glomerulus, which is a knot of capillaries inside Bowman's capsule. Blood exits through the efferent arteriole, narrower. That difference in width is what creates high hydrostatic pressure inside the glomerulus, forcing small molecules through the filtration membrane into the Bowman's capsule. This is called ultrafiltration. The cells lining Bowman's capsule are called podocytes. They have finger-like projections that form filtration slits. Now, here's something NEET has tested. Blood cells and large proteins do not cross this filter. If they appear in urine, that's a pathological condition. Know this. But urine formation has 3 stages, and what happens at each stage is exactly what NEET tests. Stage 1, glomerular filtration, 180 liters per day. GFR is 125 milliliters per minute. Everything small passes through. Glucose, amino acids, water, sodium, potassium, urea. Stage 2, tubular reabsorption. This is massive. The PCT handles about 70 to 80% of all reabsorption. Glucose is 100% reabsorbed, under normal blood glucose levels. This is active transport. Water follows by osmosis. Sodium is both actively and passively reabsorbed. The loop of Henle adds the next layer, setting up the medullary concentration gradient. DCT and collecting duct do the fine adjustment, fine-tuning water and ion balance under hormonal control. Stage 3, tubular secretion. This is the body pushing substances from the blood into the filtrate. Hydrogen ions, potassium ions, ammonia, certain drugs, they get secreted here. This is critical for acid-base balance. By the time filtrate exits as urine, it contains urea, creatinine, uric acid, excess ions, and very little water relative to what was filtered. Let's talk about regulation and the NEET traps hidden here. The Renin-Angiotensin system. When blood pressure drops, the juxtaglomerular apparatus, or JGA, in the kidney releases an enzyme called renin. Renin converts angiotensinogen in the blood to angiotensin 1, which becomes angiotensin 2, a potent vasoconstrictor. Angiotensin 2 also stimulates aldosterone release, which retains sodium and water, raising blood pressure. This is a cascade NEET has tested at every step. Another one, erythropoietin. When oxygen levels are low, the kidney releases this hormone. It stimulates bone marrow to produce more red blood cells. This is why the kidney is considered an endocrine organ, too. Then there's the JGA itself. It has macula densa cells in the DCT wall and juxtaglomerular cells in the afferent arterial wall. They sense pressure and filtrate composition and adjust accordingly. And finally, micturition. The bladder accumulates urine. When it reaches about 200 to 400 milliliters, stretch receptors trigger the micturition reflex, a neural signal that causes bladder contraction and sphincter relaxation. Voluntary control is from the cerebral cortex. And that's a wrap on day 19. Let's lock in the master numbers one more time. GFR, 125 milliliters per minute, 180 liters per day. Final urine output, 1.5 liters per day. About 1 million nephrons per kidney. Medullary osmotic gradient, 300 to 1200 milliosmoles per liter. Bladder triggers micturition at 200 to 400 milliliters. These will come in your NEET paper. I guarantee it. Day 20 is locomotion and movement. Subscribe so you don't miss it. If this episode helped you, share it with one friend who's preparing for NEET. That's the best thing you can do for them today. See you tomorrow.